Hi everybody, welcome to today's webinar. We will be beginning shortly at around 12:30 European time, so bear with us. Please do introduce yourselves in the chat, it would be great to hear from you. And just to say, we have a separate Q&A box for any questions that you may have. So feel free to post your questions for Philip or for the wider Sprint project in the Q&A. Otherwise, general discussions and chat belongs in the chat section. So yeah, it's 12:30, so I think without further ado, we'll get going. So this is our fourth Sprint webinar. So welcome everybody, it's lovely to see you all. So today's webinar is going to be presented by Philipp Mäder. So Philip is completing his PhD at the University of Hohenheim as part of a Sprint project. So for those of you who aren't familiar with the Sprint project itself, we are a 5-year EU-funded project. We're coming to an end in June, so we are well underway. We have lots of exciting findings to share, and we are assessing integrated risks and impacts of pesticides on environment and human health, and this is both at regional and European level. So our ultimate goal is to inform and accelerate the adoption of innovative transition pathways and to inform risk assessment towards more sustainable plant protection in the context of a global health approach. So I will also share some useful links in the chat shortly. So just to direct you to the Sprint website, we have a nice newsletter that's circulated every so often where you can receive further updates from us. So in terms of today's session, so as I said, Philip is joining us from Hohenheim. So Philip is working on pesticides and soil microorganisms for his PhD. So today he'll provide an overview of that research. He's been focused quite a bit on glyphosate breakdown and substrate utilisation. So he will also share some of his other experiments. I will also, again, Philip's produced a brilliant research poster and a peer-reviewed paper, which I will share in the chat shortly. So I'm very pleased to hand over to Philip, who will share some of his research with us.
Yeah, thank you for the nice introduction. I hope you can all see my screen and the presentation. And we'll begin with this webinar about pesticides and soil microorganisms. So what are soil microorganisms? Soil microorganisms are bacteria, actinobacteria, fungi, and protozoa that live in our soil, and they perform very important ecosystem functions. One example of this is organic matter decomposition, where with larger soil animals, they break down organic residues, ultimately producing CO2 and releasing nutrients. These nutrients can then also be cycled and recycled and, for example, made plant available to improve plant health. Another job that soil microorganisms perform is the suppression of pathogens and bioremediation. And bioremediation we'll talk a little bit about today. So bioremediation is the decontamination of pollutants in soils, for example, heavy metals or pesticides. So how do pesticides and soil microorganisms get in contact with each other? We all know that pesticides are applied in agriculture, and as such, this is one pathway. By direct spray application or by the introduction of seeds that are dressed with pesticide coating, pesticides can reach the soil. But there is also an indirect application where, for example, by runoff or by spray drift during the application of pesticides, unwanted areas such as organic farms, grasslands, or forests are getting in contact with pesticides. And from the picture here, you can clearly see that the soil is treated with some kind of chemical, and pesticides or microorganisms are among the first that get in touch with the pesticides. There is a variety of interactions that can happen with pesticides and soil microorganisms, and there is a nice concept of yin and yang, where on the bright side, microorganisms are able to yield nutrients and energy from the chemical breakdown of the pesticides, but on the dark side, there are toxic effects of those chemicals. And we'll talk about that a little bit further, but overall, both of these sides can lead to changes in the abundance, the activity, and the community structure of microorganisms. So let's look at the degradation of pesticides at first. As I said earlier, bioremediation is an important ecosystem service that microorganisms perform, and microorganisms are able to degrade a variety of pesticides. Both fungi and bacteria are capable to degrade those components. This degradation often happens that a parent compound is transformed into smaller and often less complex molecules. As here, for the example with glyphosate, we can see that there are two major pathways in which glyphosate can be degraded, either on the AMPA pathway or the sarcosine pathway. And the molecules get less and less complex, smaller molecules. And at the end, we have CO2, for example, and those reactions are often catalyzed by enzymes such as hydrolysis or oxidation. And microorganisms can, for example, use here in example B, the phosphorus that is cleaved from glyphosate as nutrients, or use the CO2 for energy production. On the other side, we have the toxic effects of pesticides, and similar to the example that insecticides can harm beneficial insects, we also have the case that, for example, fungicides that target pathogenic fungi in soil can also affect beneficial soil-borne fungi. Some of these fungicides, for example, inhibit the respiratory complexes of fungi, but this can also affect bacteria, for example. Other indirect effects are associated with changes to the community structure, and there has been a really nice paper where they could show that the application of pesticide changes the robustness of a community. So if an environmental change happens, this community is less likely to be, let's say, resistant or adapt to the new situation. Another outcome of those pesticide study is that there is a loss of functional diversity in the microbial community. So some pesticides affect a part of the microbial community more than another one. And this can, for example, influence nutrient cycling. So for the official tests of pesticides on soil microorganisms, the OECD has two tests, the 216 and 217, which is the nitrogen and carbon transformation test. These tests have been established quarter a century ago, and today many authors argue that those methods are a little bit outdated and that they should be replaced with more modern methods, which are standardized. And there are some that are well standardized and could be used. Another problem with those OECD tests is that they are at first limited to 28 days. And if the effect of the pesticide is not larger than 25% compared to the control, the experiment is stopped. However, many of those effects can only be seen after a longer period of time. For example, if we think about persistent pesticides, or also about the metabolites that are produced by degrading the parent component, which can be more toxic than the parent component. Other studies have found that modern methods, for example, qPCR methods, can actually detect effects that the OECD tests cannot affect. But to be fair, we have to say no method detects it all. So there are a lot of challenges also with single pesticides. Another challenge arises because in our environment there is not only one factor that affects, for example, soil microorganisms. And here we see a study by Rillig et al., where they tested single global change factors on soil microorganisms, for example, drought, but also glyphosate or salt. And we can see that while some effects are negative, we also have some positive effects in those yellow dots. And then they combined those stress factors with an increasing number of factors. And this we can see with the black dots. And we can see that by increasing the number of factors, the effect that these stressors have on the microorganisms is getting directionally more negative. Similarly, Mäder et al. From last year, they performed this similar experiment, but only with pesticides. And you can see this on the right, and especially at the bottom, where the single pesticides have, let's say, a slightly positive effect, but when combined with 5 or with 10 pesticides, the effect is getting increasingly negative. And from the Sprint field sampling campaign and other studies, we do know that there is not only one pesticide present in our agricultural fields and in the environment, but there are multiple pesticides present, also in organic fields and other environments. So what did we do? Well, you have to start somewhere, and it's always difficult to decide what you want to do, especially if there are so many pesticides present. So we aimed to investigate the effect of multiple pesticides with a special focus on microbial functions. This was done to improve our mechanistic understanding, and we performed 2 experiments, and the first one I will present first, and then a short introduction into the second one. So in a first experiment, we wanted to look at pesticide degradation as a special function of microorganisms and how this is affected if there is more than one pesticide. For this, we used 13C labeled glyphosate, which allows us to follow the signal of this 13C into the soil and into soil microorganisms. We performed a 56-day incubation in microcosms that you can see in the picture. Those are enclosed systems. And we used 3 pesticides. Obviously, we used the 13C labeled glyphosate, which is well known to many. And as a second component, we chose the herbicide MCPA, which is a model substance for xenobiotics. It's rather fast, or it's quickly degraded. And as a last component, we used the fungicide difenoconazole, which was found in, or a part of the triazole group was found in Sprint soils as well. And this is, a persistent fungicide. We used different concentrations. So for glyphosate, we used a 20 milligram per kilogram treatment, which is slightly higher than the recommended field dose. But this was used as a total load of pesticides that we have in the following treatments as well. Then we used a glyphosate treatment with 10 milligram per kilogram. This reflects the actual amount of glyphosate in the mixture treatments. Then coming to the 2 double pesticide treatments, we mixed glyphosate and MCPA or difenoconazole, both with 10 milligram per kilogram. And for the triple treatment, we again used 10 milligram per kilogram of glyphosate and each 5 milligram per kilogram of MCPA and difenoconazole. For all of those pesticides, we determined the residues during the incubation. So we took samples at several time points. And what we could do with the microcosms that we used, we could measure the CO2 evolution by a head trap or headspace trap in those containers. And this is usually used for an indice for activity of microorganisms. As we labeled the glyphosate with 13C, we could also determine the 13 CO2 content, which is indicative of a complete mineral or a mineralization of glyphosate. Then we measured the abundance of microorganisms by utilizing microbial biomass carbon. And again, this carbon has some sort of 13C label, which is indicative for the utilization of glyphosate for growth. Then we also wanted to know which of those microorganisms utilized glyphosate and how it changes the community structure. For this, we perform the phospholipid fatty acid analysis, which allows us to determine between gram-positive, gram-negative bacteria and fungi, and also with the 13C label, which of them utilized glyphosate. As I said earlier, enzymes are involved in the degradation of the pesticides, and fairly recently there were actually genes developed or found that are involved in the pesticide degradation. So these genes encode for the enzymes that catalyze those pesticide degradation reactions. For example, for glyphosate, this is COX-A and SARC, and for MCPA, it's the TFD8 gene. We were also able to calculate some indices. One of them is the metabolic quotient, which is often used in ecological studies, which gives us an idea how much carbon is respired per biomass carbon. If this increases, it is usually associated with a stress reaction of the microorganisms. Similarly, we could do that with the amount of substrate-derived carbon that is used for growth in relation to the total amount of 13C that is respired and taken up in the biomass. So let's jump into the results. Here you can see the respiration rate on the different incubation days. And at the beginning with the black circles, we could see that the presence of MCPA, either only with glyphosate or in the triple treatment, increased the respiration rate initially. On the other hand, with the red arrows, we could see that after day 28, difenoconazole decreased the respiration. And we'll come back to MCPA in a second. We'll start with difenoconazole further. On the left side here, you see the residues of difenoconazole. And unsurprisingly, as it is a persistent fungicide, the residues did not decrease significantly over the duration of the incubation. However, if we calculated the cumulative CO2 production after 56 days, we could see that difenoconazole decreased the respiration or the cumulative respiration after 56 days. This has been observed for other fungicides of the triazole group. However, those triazoles usually inhibit a membrane component of fungi named ergosterol, so there's no direct effect on the respiration. And we could not observe a decrease in fungi or bacteria, so difenoconazole somehow is able to also influence the activity of microorganisms. Moving on to MCPA, also unsurprisingly, it is degraded quite rapidly, and this could also be shown on the left with the residues. It was degraded within the first week, and we could also see that there was a concentration-dependent effect, where on day 7, the higher initial amount of MCPA was still visible in the residues. On the right side, you see the abundance of the TFDA gene, which is involved in MCPA degradation. And we can see that this only increased on day 7. So this increase must have happened somewhere between day 2 and day 7, where a microbial community established that is able to degrade MCPA. And after day 7, the abundance of this gene decreased, but it was still visible after 56 days. Nevertheless, the main degradation of MCPA happened somewhere between day 2 and day 7. As we saw earlier, MCPA initially increased the respiration of the microorganisms, but at the same time, there was no change to the abundance or the microbial biomass carbon. This leads to the metabolic quotient increasing, and this means that there is some kind of stress reaction. The initial increase in CO2 is also likely from the MCPA carbon, so the degradation of MCPA producing CO2, which increases the overall CO2 levels. However, as they did not use it for biomass production, most likely, this indicates a stress reaction. Now moving on to glyphosate. Here you see the amount of glyphosate that has been mineralized during the incubation as percent of the initially applied glyphosate. And we can see that the patterns are quite similar during the incubation, and especially in the mixture treatment, in the triple treatment, which is the one on the far right, we have an increase of up to 40% in the degradation of glyphosate. This has to do with the presence of MCPA, because in the MCPA treatment, which is the second from the right, we can also see that there is an increase in glyphosate mineralization. So how can this occur? There was a study that suggested that MCPA can increase bioavailability of glyphosate, and there are two main ideas of how this can happen. So if we imagine that glyphosate or MCPA are adsorbed to mineral surfaces in the soil, so glyphosate on the left, MCPA on the right, and the other component is in solution. What could potentially happen is that on the left side, MCPA exchanges glyphosate from this mineral surface, thereby increasing its bioavailability for mineralization. Or on the right side, that MCPA blocks binding sites of glyphosate, also leaving it in solution, increasing its bioavailability. Up to now, we only talked about single effects or double effects, but what happened in the triple treatment? For the cumulative respiration, we saw that difenoconazole decreased the cumulative CO2, while MCPA initially increased it, and in the triple treatment, we have some sort of a compensatory effect. Both pesticides are present, plus glyphosate, but the effects cancel each other out. And on the right side, with the glyphosate mineralization, we could see that MCPA and glyphosate treatment increased the mineralization of glyphosate, and the one with difenoconazole, which is in the middle, only slightly increased its degradation. If difenoconazole and MCPA are both present with glyphosate, this mineralization is even higher than only with the MCPA treatment, meaning there's some kind of cumulative effect. And we could further see that in the carbon use efficiency, where we have a decrease in carbon use efficiency, which is somehow in line with the triple treatment. So in this treatment, the effect is stronger than in the single pesticide treatments. Also, this means that in this triple treatment, the energy demand of the glyphosate degrader community is increased while their growth is limited. So they rather respired the carbon from glyphosate than use it for biomass production. Our conclusions from that first experiment were that the interactions of the pesticides that we used can lead to both additive, so cumulative, and antagonistic effects on soil microbial parameters. At the same time, it is not only the number of pesticides that determine the effect, but also the individual effects that they have. As we've seen, MCPA increases the respiration at first, but difenoconazole decreases it later. All the effects were stress reactions and changes to the potential for glyphosate degradation and alterations to carbon utilization patterns. In a second experiment, which is not published yet, but there's a preprint already available, we wanted to see how pesticides alter substrate utilization. And for this, we used maize leaves, as they have a naturally increased 13C content that we can again use as marker and follow its fate. We also wanted to see how substrate quality plays into this. And for this, we either used fresh maize leaves that are cut into small pieces, or we artificially age them. That you can see in the picture, it looks like tea, it isn't. We soak those maize leaves in water for 72 hours to decrease the amount of easily available carbon. Then again, we incubated it for 56 days with 2 different pesticides, one being the fungicide boscalid, which was also frequently detected in spin soils, and the other one is prosulfocarb. This is a herbicide that affects the lipid synthesis in plants, and we hypothesized that this pesticide could also interfere with the lipid synthesis in microorganisms. So I'll only quickly go over the results, and starting with the residues of prosulfocarb. In green, you can see the residues on the maize leaves, and in dark brown, you can see the residues in the soil. These are the prosulfocarb residues, and they decreased rather rapidly on the maize leaves, which is in line with some observations that organic matter can enhance the degradation of a pesticide, for example, by delivering more nutrients or by co-metabolic reactions. We could also observe that in soil, we have a lag phase of about 14 days. This is also, or was also observed for hydrophobic pesticides such as prosulfocarb. The more interesting results were with the boscalid residues. While we could see a steady decrease on the maize leaves, we can at the same time, we can see an increase in the residues in soil, which means that there is a transfer of the boscalid residues from the maize to the soil. At the same time, the pictures on the right, the 2 tiles, are the combination treatment between boscalid and prosulfocarb. And we can see that there on the maize leaves, there is some kind of a lag phase of the reduce of boscalid residues, which is likely associated with a preferred mineralization of prosulfocarb at first. After prosulfocarb is gone, they can start to degrade the boscalid. Now quickly jumping into the effects on soil microorganisms with prosulfocarb. As I said earlier, we hypothesized that the mode of action of prosulfocarb by inhibiting lipid synthesis in plants can actually also affect soil microorganisms. And this was the case for bacteria. We could actually see that the new formation of phospholipids in biomass was inhibited with prosulfocarb. So there's a non-target effect on bacteria. Further, we could show that there was a reduce in carbon use efficiency of the maize leaf, so there is a stress reaction, and we could also observe that there are some shifts to carbon limitation and enzyme activity. For boscalid, it was again a connection with the mode of action of this fungicide. Boscalid decreases the respiration and maize mineralization in our experiment, and this has to do with its mode of action, and it acts as an SDHI inhibitor, which is a part of the respiratory complex in fungi, and we actually also found a decrease in fungal abundance at the beginning of the experiment. Also fairly unsurprising, as a fungicide, it can also affect non-target fungi. What we could also see is that in the fresh maize treatment, after an initial decrease of the fungi, we could see an increase in fungi again. So there are different communities of fungi that are reacting to maize, to the pesticides, but they are also affected by the substrate quality. So from this experiment, we concluded that there is potentially a transfer of pesticide residues from plant materials to soil. And this could lead to an accumulation of residues in soil that is not calculated in residue predictions or in modeling, especially for pesticides that are somehow persistent or hydrophobic. that can get transported with the organic material that is left on the field and then incorporated into the soil. But we could also show that the mode of action of the pesticide can affect non-target organisms. So to conclude, overall, pesticide mixtures can alter the effects of single pesticides, and this makes it really hard to predict the effects. On a timescale, we could show that effects are still visible on day 56 or only happen after day 28. This is especially true for persistent pesticides, so official tests need to think about this. And what is really important, I think, is that real-world mixtures need to be included into official tests. And with that are my references, and I thank you for your attention.
Fantastic. Thank you so much, Philip. That was a great presentation and presented really clearly. So we'll get underway with a Q&A. Oh, great. It's good to see the audience clapping. Yeah, no, I hope you all agree that Philip did a fabulous job. So we will get underway. Oh, more clapping. Brilliant. We'll get underway with a Q&A session now, because I know we've already had several questions, which I hope Philip will be able to answer. We also have Vera from the Sprint project here, who may be able to answer any questions that you have about the Sprint project itself. So just to get started, we had a question from Bill Grayson, and I think this is aimed at Philip. So the question is can bioremediation also reduce microplastic burdens in soils? I appreciate this isn't your research area necessarily, Philip, but I wondered if you had anything you could say on that.
Well, yeah, like you said, it's not my field of expertise, but I had a look at microplastics just out of interest. So as far as I know, microplastics are not really degradable by microorganisms. There are some cultures that have been isolated that are able to degrade microplastics, but if this is present in our environment, I'm not sure about that. And if you could inoculate, for example, our soils with those species, let's say, yeah. But that's all that I basically know about that. Yeah.
Yeah, no, perfect, Philip. Thank you. And if you're interested in the impacts of microplastics on soil health, you may also want to refer to another EU-funded project, Minagris. So I'll post in the chat briefly. But Minagris is looking directly at micro and nanoplastics in agricultural soils. So Bill, that might be a good way forward. So I have a question from Nathan Einbinder, who posted actually in the registration form before this webinar. But he asked, is it possible to speed up recovery of the soil microbiome after pesticides? So possibly over many years and decades by, for example, adding biological inputs or fermented native microorganisms. So yeah, I guess this is around, what happens after pesticide use? How quickly can you recover the soil microbiome?
Yeah, well, while fermented native microorganisms doesn't say anything to me, I'm not sure what you mean by that. Maybe you can clarify that. It's definitely possible, depending on the effects that the pesticides have on the microbial community, to help them recover with some biological input. It depends on how the microorganisms are affected and also what your aim is. There might be irreversible effects that the microbial community will never recover as it was before. And yeah, there's definitely ways. There has been a study also recently about the effects of also multiple pesticides where nitrogen addition actually hampered or mitigated the effects of the pesticide. So there's definitely a way to improve the nutrition of microorganisms to recover. The time span, that is a subject for probably long-term experiments that have not been established yet or have not been tested yet. And like I said earlier, I think that the microbial community will never come back to the state that it was in a natural ecosystem. Oh, yeah.
Brilliant. That's perfect, Philip. Thank you. So a question from Jan Tien. So they have asked, for the bioavailability side of things, have you taken into account the effect of binding on dissolved organic matter?
So for the bioavailability of glyphosate, the mineral surfaces was just an example. It could also happen on organic matter, obviously. And as we've seen for the second, or with the second experiment, where we actually have this binding of the pesticides to the maize leaves, this is the case, yes. Yeah, so it has been taken into consideration, but we did not test for it, or yeah.
Yeah, perfect. And Jantine also asked, she also asked pH is an important factor in bioavailability. Did you take this into account, as different soils have different pH?
So we chose a soil, especially for the second experiment, which is in line with the OECD standards, with a pH that is, I think, slightly acid, but close to neutral. But this was taken into account, obviously. Like I said earlier, you have to start somewhere, and especially the statement different soils have different pHs. Yes, that is true. Also has different size distribution, particles, organic matter content, water content, etc. There are so many factors that can influence pesticides further. So, and this is, I think, also really challenging for all scientists and regulators, and that's why the OECD has some kind of norms put in place, or yes, because they use a very specific type of soil, which they describe would be the worst case scenario of a soil with slightly higher sand content. And yeah, it's challenging to find a soil that you cannot, what's it called? You can't upscale such experiments. I think it's proof of concept. It's trying to understand some mechanisms that could happen. Yeah, definitely a lot of factors influencing all these interactions.
Yeah, no, absolutely. You're completely right. You know, of course, soils have many, many different features, pH being one of them. And as we all know, especially if we're on farm or anything, yeah, soils vary an awful lot. So it does make it quite tricky. Maxime Warnow asked before the webinar, how should we choose which pesticides that are present in soils to study when we're looking at their impact on soil microorganisms? How many should we select? Considering that the more we study, it might make it more tricky to be accurate. I guess, yeah, this is around, you know, how did you decide which pesticides to look at?
Yeah, really interesting questions. The question that also took some time when we started the experiment. I think it always depends on what your aims are. Like I said, we wanted to understand the mechanisms behind some of those interactions. So we chose pesticides that are, or two of the pesticides that are well studied. And also, like I said, at the end of the presentation, it's really important to study pesticide mixtures that are used in real scenarios. And I think from this you could have further or more information, and you could have more implications for also regulators, for example. Yeah, I think on how many pesticides you should use, well, all of them, but that's not feasible in most cases. So you have to choose, because obviously also the number of samples goes up exponentially if you introduce one more. So time, money, all determine how many you will use. You could, and also if you only want to see what the mixture does, do you want to see what individual combinations do? So there's a lot to take into consideration, and I think there's no clear answer to that.
Yeah, no, absolutely not. It's really complex. And I mean, for our audience, if you look at some of our wider Sprint publications and resources, Sprint has been looking at pesticide mixtures in various guises. So it'd be great to, check out some of our other research. So Mary Eliza has asked, well, she says, first of all, thank you for your amazing presentation. But she was wondering, while many pesticides have an antagonistic effect on the microbial community, are there some which might have a positive effect? Do you know if there have been any studies that focus on the effects of these pesticides on the phage fractions of the soil? So it's kind of a double question there, but yeah, really, really interesting stuff, actually. You know, are there any antagonistic effects?
So I think it always depends on what you describe as a positive effect.
Mm-hmm.
There are some studies that showed that there is an increase between different, or an increase in interactions of some microorganisms. So the community has more interactions, the network is getting more complex. But is that positive? Is that negative? Also, if you say, well, the microorganisms are more active, they produce more CO2, or they degrade more organic residues, is that positive in the long term? Or do you just accelerate the depletion of nutrients in the soil because the microorganisms are stressed? On the other hand, microorganisms might grow on this. Yeah, what was the second part of the question? I forgot.
So the second part was around whether there's been any studies that have looked at the effects of these pesticides on the phage fractions of the soil.
Not sure at the moment. I'm not.
Yeah. That's fine.
Yeah.
I mean, that's something that we can get back to you on. So, you know, if we are unable to answer any questions immediately today, we can reach out to you with a summary after this webinar, and we can have a look into this. Let's move on to another question. So this is from Carolina. Sorry, Philip, you're doing so well. This is almost like a Bible, but you're doing really, really well. And I think it just reflects how interested our audience is. I think it's really great stuff.
It's really good, yeah.
So Carolina says, so is there a study on glyphosate and the soil microbiome that also takes into account additional substances contained in commercial products? So I think she's talking here about potentially adjuvants, and, you know, for example, glyphosate is quite different to Roundup, you know, as sold as a commercial product.
That's actually something that I wanted to dive in a little bit deeper in the introduction at first, but I think this subject is very difficult to discuss because the formulations are often an industry secret. So it's a scientific black box, you could say. I tried to look up and find some studies on testing glyphosate alone, and for example, as Roundup. There are obviously some studies out there that use the commercial formulation. The effects might be different, and yeah, there's a lot of stuff probably in those formulations that can also affect microorganisms and maybe also affect them more than the actual active substance of the pesticide itself. But for this, the industry has to give out the secrets of their formulations so that we can test that as well. And I think this will be a debate that is hard fought.
Yeah, no, absolutely. Yeah. Yeah, I think there's a lot here that we should talk about in great detail. But I think, so moving on to another question. So this one is from Martin Bujeman. So he says, how do the findings from sort of controlled lab experiments translate to real world agricultural settings? So again, I think we're talking here about the various soil types, climates, crop rotations, you know, what further research would be needed to make sure that, the experiments do translate into real life practical settings.
Yeah, like I said before, upscaling is not allowed. This is a proof of concept. Again, it's a mechanistic idea. You have to test it in the field, and there's no answer. This is done for many studies that upscaling is done where upscaling is not possible. I mean, we had, like you said, controlled conditions. And in real life, there's a lot going on. There's rain, there's weather. Often also plowing or other things happening. So no upscaling.
Yeah, absolutely. Vera, would you also like to step in here?
If I may, because I do think this is a really, really interesting question. And indeed, like here, the work of Philipp is crucial to understand the mechanisms. But indeed, and the upscaling brings challenges on itself. But I think it's also interesting to stress that we have also looked into the current situation. So in the Sprint project, we have also we analyze the soils across different locations in Europe, and we know the mixtures of pesticides that are there, and we also look into the microbiome in these locations. So again, it's harder, of course, and it's not suitable for exploring mechanisms as Philipp is doing here, but gives us also an impression also of what real life conditions we have in our agricultural settings. Those results on the microbiome, so microbiome across Europe are not yet published, but very close. So might come also to this webinar series, maybe. But yeah, so we have looked into that as well. So keep tuned, because there is definitely something real field context on this.
Yeah, brilliant. So I think I don't see any other questions coming through. Some really interesting discussions continuing around glyphosate versus commercial products. So Ulrich is explaining how actually, you know, both have to be tested for registration, and the lowest, most critical endpoint is taken for risk assessment. Are there any further questions? If so, please do shout now. Otherwise, we would like to let you know that we are holding a Sprint final conference in June. So please do save the date. It's taking place on the 25th of June in Brussels. So it will be a hybrid event, but we'd like to encourage people to join us in person. So we will be sharing a registration form via our website in due course. So if you're keen to come, please do sign up for our newsletter updates, because we'll circulate it to our emailing list. If you agreed to be part of the list on the registration form, no need to do so. We will add you ourselves. So just to say, we are also holding a further webinar. So it's taking place on the 15th of April, I believe, and we will be hearing from Meiki Giretzi about her research, which is all about the gut microbiome and pesticides. So that's going to be really interesting. So please do join us then. We will also be posting this webinar to our YouTube channel. So if you have any colleagues who missed the session today and would like to catch up, please do direct them there. I'd like to give a really big thank you to Philipp. He's done an excellent job. You know, we normally have 2 speakers for these webinars just to take the pressure off a little bit. So he's done a great job in doing this for himself. So a big thank you. And yes, we'll look forward to seeing you all very soon. And please do stay in touch if you have any further questions. So yeah, okay, I think we'll end a little bit sooner than expected, but I think that's a good thing. We all want to have our lunch probably. So yeah, amazing. Thanks everybody. Thank you.
Yeah,
Bye.
thank you. Bye.
Thank you. Bye.